I wanted to add a little bit to what part one of this chapter, add a little bit to that. I have included other features on the Blackboard class page. If you look in, I believe it's the tab Cells, Extra Material on the cell membrane organelles. In there is an MP3 on the cell membrane and the things that you find in the cell, so more information in more depth in terms of the cell membrane, but also more information on the active and passive ways that things get into the cell. And so those are good things to give a listen to because they go into even more depth than what I'm doing here in the lectures. And more depth is always good because what happens if you can study in depth as much time as you have, then you remember at least the basic points. So there is a psychology in that, in the fact that if you go in more detail on a subject, you remember at least some of the basic information. And that's what we're trying to get in your head, is the basic information about these topics. So have a look on that. Not only is there's an MP3, which is a very portable file, which is nice, but also links to different films that are in your library streaming video service to look at, to have a kind of a different view of the material that I'm talking about here in this portion of the video lecture. The next part that we want to talk about is the cytoplasm, which we kind of been talking about all along. And what this list of material here is comparing external, internal. Cytoplasm, what's inside the cell, cytosol composition in comparison to the fluid that's outside the cell. You have dissolved materials inside the cell. There's not very much that's just hanging out outside the cell. If it goes by, I know I say that it goes by and the cell brings it in, but there is a little bit more organization than that to it. And so there is not an overabundance of things outside the cell. You have dissolved materials inside, nutrients, ions, proteins, also waste products, things that the cell is getting ready to go out, to send out. In comparison, the inside of the cell is higher in potassium and lower in sodium compared to the outside. And inside is higher in proteins compared to the outside. High carbohydrate, low amino acid and fat. You do have this carbohydrate inside so that you have a good source for, and this is comparatively. Don't think of a big bowl of pasta high carbohydrate. We're talking about minute amounts of all of this, but you have the ready source for energy. You also have some amino acids, because you do have to have a storehouse of amino acids if you're going to make proteins, your cells. Inside the cytoplasm, you have these things called organelles, small organs. They are not able to live outside the cell, but they generally have specific functions all of their own. They do a function for the cell that another organelle does not do. That's how they get their names. They can have a membrane or a non-membrane, and the membranes are very similar to that biphospholipid layer. Some of them, the membrane is integral. It's very important that membranes stay in and wall that organelle off from the cytoplasm. Other organelles that have membranes, like the Golgi apparatus or the endoplasmic reticulum, have the ability to actually create things that move through the cytoplasm. So there is more of a give and take than the other membranous organelles. And then you have the non-membranous cytoskeleton, microvilli, centrioles, cilia, ribosomes, and those enzymes that break down proteins, proteasomes. Membranous organelles are endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and mitochondria. Talking a little bit more about the cytoskeleton, we did a little bit already. It gives shape and structure to the cell. Cells eventually, when we talk about tissue, we'll talk about different cell shapes and how the shape of the cell gives the basic structure for the tissue that is created, and that tissue has a form or an architectural structure to help it or to facilitate or make the physiology that tissue does, like the skin or like the muscle, like the nervous tissue or connective tissue. In making up the cytoskeleton, you have different types of structures. You have microfilaments, and these are found mostly, well, first of all, they're composed of the protein actin. Actin and myosin, we put those two together for a muscle cell, and we'll talk about that when we talk about muscles. But the microfilament are made of the protein actin, and we mostly find microfilaments just inside the plasma membrane. So you have these microfilaments, micro based on their size, they're smaller, and they also tend to be found right inside the plasma membrane. Intermediate filaments, mid-size between micro and thick, and all of the filaments are used to maintain shape, stabilize organelles, and stabilize cell position. But intermediate filaments are found further in the cell, making up the cytoskeletal portion, more toward the middle, I guess, but not as close to the middle as the microtubules. And these are hollow. They are a different design than the filaments. The microtubules are unique in the fact that they give rise to the centrosomes, and so they're associated with the centrosomes. Centrosomes will be very important when the cell gets ready to replicate itself, go through mitosis. So they give strength to the cells. They also have this ability to move vesicles along. Vesicles are those items that are made to either move something from one organelle to another or move something from an organelle outside of the cell. So these microtubules allow that movement to take place. They will eventually form the spindle apparatus that attaches to the individual chromosomes and pulls them to different poles when this cell is dividing. And then thick filaments, which are made of myosin, are found only in muscle cells. And again, thick filaments, thin filaments, myosin and actin get together to form the functional unit of a muscle cell. Here you have a nice picture. Again, this picture comes from your book. You see the microfilaments. They're just inside the plasma membrane. And you also see the intermediate filaments, the microtubules with a vesicle associated with that. A couple of other things that we're looking at here, microvilli. Microvilli, this increases surface area. And then you have a centrosome, which is the area around a centriole, and this will be associated with mitosis. Cilia are larger than microvilli, and cilia allow for a movement. You see cilia in the respiratory system. You also see it in the reproductive system. It facilitates movement of something. In the respiratory system, it facilitates movement of larger particles out of the respiratory system. And so you do have the capacity to have a motility, so to have movement with regard to the cilia. There's also another type of cilia called a primary cilia that acts kind of like a antenna to pick up information, in a very small area, so among just a few cells, data about external environment around those individual cells. Ribosomes, these are, again, we're talking about the non-membrane organelles. They are the sites of protein synthesis, so building polypeptides. So they are the sites where you're putting amino acids together into polypeptides. You have two types. You have free ribosomes, this floating around, well, actually moving around the cytosol, and then fixed. Fixed ribosomes are attached to the endoplasmic reticulum to further facilitate. So free ribosomes would be part of the beginning of putting the amino acids together. Fixed ribosomes further facilitate that design of a protein. Remember, the protein has the primary, secondary, tertiary structure, and then on to even a quaternary structure. So you have the primary structure, and then moving into actually designing this protein into something that is has 3 dimensions and is unique or perfectly designed. Then you because protein design can be complicated, and sometimes the body doesn't get it right, or sometimes proteins need to be eliminated, you have an enzyme, protease, and it will actually recycle proteins. So disassemble them, take them apart. Those parts can be used again, but that particular protein wasn't created, or it's lived its life. It's basically needs to be destroyed. Again, sorry to cut this off, but it says endoplasmic reticulum there at the top, ER, endoplasmic reticulum, and you have a little bit of an image here. Endoplasmic reticulum is made up of, we tend to call this the, you have these, I guess, flattened membranous areas where you have cisternae, and those flattened areas where you have little openings or storage areas within the membrane. So it's a membranous organelle with storage areas within, and the endoplasmic reticulum is for synthesizes proteins. So in addition to, so the next place that proteins go after the ribosomes, carbohydrates, lipids, storage of molecules, and materials for transport within the endoplasmic reticulum. Transport within the endoplasmic reticulum, because there is there are many steps to protein synthesis, and also carbohydrates and lipids. This is also where particularly some drugs and toxins are detoxification, so neutralized. Endoplasmic reticulum comes in 2 types. You have smooth if there's no ribosomes, and rough if ribosomes are attached to the surface. So smooth with no ribosomes functions to synthesize lipids and carbohydrates, phospholipids and cholesterol. One of the big functions of the smooth endoplasmic reticulum is to fix the to create the fixture for the plasma membrane, and also membranes of other of the cell or organelles. Also create steroidal hormones, glycerides, and glycogen. So glycogen is a stored form of energy in the muscle. Glycerides are a stored form of energy in the liver. Rough endoplasmic reticulum, what you have there is the further manufacture of proteins, and then putting those proteins in transport vessels. We will continue in the next one.